What's up, subscribers? Today we're going to be talking about protein synthesis. Protein synthesis starts off with DNA according to the central dogma of living things. DNA is created with the base pairs of adenine paired with thymine and cytosine paired with guanine. DNA is created in a double helix structure and is used to carry the genetic information of all living things. To begin transcription, the first process in protein synthesis, transcription factors in the nucleus begin to prep the DNA to be transcribed and will bind to the TATA box located in the promoter region of the DNA coding strand. RNA polymerase, an enzyme responsible for creating RNA strands, will then begin to build the RNA. The 3' to 5' strand will be used as a template strand so that RNA polymerase can build in a 5' to 3' direction. RNA polymerase will begin to assemble nucleotides to create a new strand by reading the DNA strand. The strand will be nearly identical to the coding strand, but uracil will replace thymine. Once the new strand is done, it will become a primary transcript. RNA polymerase will run into a termination sequence at the end of the gene that will signal to stop transcription. The RNA polymerase will then detach from the DNA sequence. Before the strand can move on to translation, the pre-mRNA strand must undergo modifications. A 5' methyl cap is added to help the strand get through the nuclear pores, and a 3' poly A tail is added to prevent the RNA molecule from losing information. Spliceosomes splice out intron sequences from the mRNA, which are non-coding regions, and leaves exons, protein-coding regions. Now we have an mRNA strand ready to go into translation, the second step in protein synthesis. After the mRNA travels to the cytoplasm from the nucleus, the mRNA strand will then travel through the ribosome to create a polypeptide. For translation to begin, an initiator tRNA molecule will need to bind to the 5' end of the mRNA along with a small subunit of the ribosome using the 5' methyl cap. The mRNA will then be read 3 nucleotides at a time, referred to as a codon. They move along the RNA in the 5' to 3' direction until the start codon AUG is reached. The LARP subunit of the ribosome will attach, and the mRNA is now in the P site of the ribosome. For each codon, a tRNA molecule with an amino acid matching that codon will attach to the codon. That tRNA molecule must be an anticodon of the codon it is trying to attach to. Once attached, a peptide bond will form between the amino acids. The cycle will be continued with mRNA being pulled to the ribosome a codon at a time. Translation will stop when a stop codon such as UAA, UAG, or UGA enters the A site. Proteins called release factors in the P site recognize the stop codons and add a water molecule to the end of the polypeptide chain. The small and large subunit of the ribosome will disassemble and end translation. The polypeptide will then enter a chaperonin to become a folded protein and complete protein synthesis. That is all for protein synthesis. Make sure to subscribe and like our channel.